Bonjour à toutes et à tous. Alors, bienvenue au séminaire du groupe Énergie-Climat. Alors, si vous ne connaissez pas très bien ce groupe, je vais vous mettre le lien vers le site web, de la page web. Et vous pouvez aussi vous abonner aux listes donc newsletter Énergie-Climat et groupe Énergie-Climat si vous voulez suivre les activités. Donc, je prends la suite d'Emmanuel Lelouch pour l'organisation des séminaires. On va partir sur une fréquence de 2 séminaires par an. Et pour aujourd'hui, on souhaitait entendre parler d'océans et de biodiversité, des sujets qu'on n'a pas tellement traités jusqu'à présent, et avec en particulier le rôle fondamental joué par le plancton pour l'atmosphère et le climat, puisqu'il est à la fois source principale d'oxygène et puits de carbone. Alors, on est très heureux d'accueillir pour en discuter Chris Boller. Chris Boller est directeur de recherche au CNRS depuis 2002. Il dirige le laboratoire de génomique des plantes et des algues à l'Institut de biologie de l'École normale supérieure de Paris. Il a reçu de nombreux prix très prestigieux pour ses travaux. Je vais juste souligner qu'il est, depuis cet été, professeur au Collège de France et il donnera sa leçon inaugurale la semaine prochaine, si je ne me trompe pas, le 4 février à 18 h, sur la biodiversité et les écosystèmes à travers le temps et l'espace. Donc, si vous avez envie d'en apprendre plus après ce séminaire, n'hésitez pas à aller suivre ce cours. Donc, ces travaux de recherche portent en particulier sur la compréhension de la réponse des plantes et du plancton aux signaux environnementaux par de l'analyse génomique. Et depuis 2009, il est l'un des coordinateurs scientifiques du projet Tara Océan, dont il va beaucoup nous parler, et il est même maintenant directeur scientifique de Tara Océan. Et il a été l'un des directeurs scientifiques, notamment de l'expédition Tara Oceans Polar Circle. Donc, on part sur un séminaire d'environ 1 heure. Alors, Chris parle très bien français, mais pour plus de facilité, le séminaire sera en anglais. Et ensuite, on aura du temps pour des questions que vous pourrez poser en français ou en anglais, sans doute. Donc, thank you a lot, Chris, again, and please, I let you the stage.
Okay, thank you very much, Benjamin. Do you hear me okay?
Yes, perfect.
Okay, great. Okay, so it's nice to interact with you in the Observatoire. I know that we are neighbors almost in Paris. I see you from far when I walk to the École normale supérieure, and I think of what you're doing in the Observatoire on the Boulevard Saint-Michel. So thank you for the invitation to present our work to you today. Most of what we do is really biology, so I don't think there are so many biologists in the audience, so I will not go into too much detail, but I will hopefully try and give you an idea of what we can understand about how the ocean functions using different approaches to study the biology that is present in the ocean. So I will do that using the Tara Oceans project as example. If you didn't hear about Tara Oceans began with a 4-year expedition on the research sailboat called Tara, that you see on the right side. This is owned by the Fondation Tara Océan, which is part of the Agnès B group. Okay, so Agnès is very sensitive to environmental issues, and she graciously gives some of her own money to support the foundation's activities, which happen through the interaction with scientists like ourselves to perform long-term research expeditions using the Tara sailboat to understand new things about ocean processes. So in our case, the Tara Oceans project was really focused at trying to understand the planktonic ecosystems in the ocean, which I will talk about during this presentation. But there were also some major activities of the foundation in particular, around the popularization of science towards the general public, the education of children in the schools, and also to influence governance policy related to managing the ocean, okay, which we try to do through numerous activities. Today, I will only talk about the scientific component of the project, but if you want to know more about what we have done in terms of popularization, education, policy, issues related to science of the oceans, and I can happy to talk to you about that later. As I say, the project began with a 4-year expedition. I will tell you a bit more about that later. And the project was run, was really inspired by Eric Karsenti, who was a cell biologist and also a sailor, who really inspired us to initiate this project. He was at that time head of department of the EMBL, which is the European Molecular Biology Laboratory in Heidelberg. This is the biologist equivalent of the CERN, which I'm sure you've heard of, which is a European grouping of people focused on using molecular biology approaches. The CNRS was also very involved in the project, and also the CEA, the Energy Agency of France, notably through participation of the Genoscope Sequencing Center in Évry, just south of Paris. So, as always in scientific expeditions to explore the oceans, we use different kinds of techniques to try to do that. Ocean exploration is a few centuries old. In the past, we had fairly simple techniques to explore the ocean using plankton nets, using microscopes once they were invented. And this led us to some general ideas of how the ocean works. Today, of course, we have much more powerful approaches to study the ocean. In addition to oceanographic ships, such as the Marion Dufresne, the Pourquoi Pas from Ifremer, we also have potential to use smaller research vessels, including sailboats like Tara. In addition to those, we have equipment that we can deploy in the ocean that goes down to the bottom of the ocean to pull out samples. And we also have autonomous vessels, such as floats and gliders and autonomous vehicles that we can also use, program to explore specific processes in the ocean. And of course, today we have satellites which are observing our planet and also bringing us very important information about the ocean. So we can study the ocean, in fact, without even getting our feet wet. We can do it now from the office using this sort of data. Our project was, however, very wet and involved a lot of research on Tara at sea. And we were focused then specifically on studying this compartment of life in the ocean called the plankton. Plankton are defined just simply as organisms which drift with the currents. But we can consider them as the invisible multitude of life in the ocean because they represent around 2/3 of the biomass in the ocean. So if you would take a giant net and scoop up all of the organisms living in the ocean and just see where the carbon is partitioned in those organisms, we would see that 2/3 of that carbon is in these microscopic organisms. The other 1/3 is in the fish, the dolphins, the whales, you know, these things that you see on TV. But in fact, the majority of biomass in the ocean is microscopic. However, even though it's microscopic, it has very important activities. The plankton are the base of the oceanic food webs. So if there is no plankton, there is no fish because they have nothing to eat. The photosynthetic kinds of plankton, they take carbon dioxide out of the atmosphere and they generate biomass, exactly like plants do on land. And in the same way as plants, they also generate the oxygen that we breathe. So this is in fact as important in the ocean as on land. It's a very important process that happens in the ocean, even though we cannot see these plants in the ocean. They are as important as the plants on land, quite amazingly. They're also important for regulation of the climate, as I will come to a little bit later, with in representing the biological carbon pump, whereby carbon dioxide from the atmosphere can be sequestered in the ocean interior for geological periods of time, and thus contributing to climate regulation. So these are sorts of reasons why we wanted to study plankton, to learn more about them using this expedition. As you see in this mandala behind the text here, there's a lot of different organisms present in this microscopic world. So let's just briefly look at these different groups of organisms before we look at specific detail. So one group of organisms that we have in the plankton are known as protists. These are quite complex cells. They're as complex as the cells in our body, which are called eukaryotic cells. They have DNA inside the nucleus within the cell, and there's a lot of general complexity of these cells. You can see in this image a lot of different morphologies of organisms, even though all of these are single-celled organisms of just a few microns in size. They have these amazing architectures. Some of them look like boxes, some of them look like alien spaceships, and so on. So these have one group of organisms that are very important that we can see in the ocean. If you take the average liter of seawater, we can see that these organisms can be present at up to 10 million cells in a liter of seawater. Okay, so you already begin to see how many cells there are in this microscopic world. In addition to these protists, we also have zooplankton, which are multicellular. So just like we are multicellular, we have different kinds of multicellular organisms in the ocean. They again have different morphologies. Some of them look like small, shrimps, crevettes that we call copepods. Some of them look more like snails and so on. And again, these are abundant in the ocean and can be present at as many as 100 or so organisms per liter of seawater. Okay. Then in addition to these, we also have lots and lots of bacteria, prokaryotes, which again do all kinds of important things, including photosynthesis, but not only. These can be present at up to 1 billion cells in an average liter of seawater. And then we have lots of viruses. You all know now of coronaviruses. There are lots of viruses out in the natural world. Most of them are not toxic and not infective for us. In fact, they are in the environment to infect other organisms. In the ocean, again, we have lots of viruses, up to 10 billion particles of viruses per liter of seawater. Okay, so here you begin to see how all of this biomass comes in the end to represent this 2/3 of total biomass where we find carbon in the ocean. So we see that these organisms are present in different numbers. They're also present in different sizes. Here we have an image of a zooplankton, which is the copepod, with a protist, in this case a diatom sitting on it, and the bacteria sitting on the diatom. Okay, so we start with the copepod, then the protist, and then finally the bacteria sitting on the diatom. So here we don't have the viruses. If the viruses were present, they would represent, about the size of my pointer, around a pixel, So you see from this video that we're going through lots of different orders of magnitude of size of organisms. So this is a big problem already if you want to study these organisms. And in fact, it would be the equivalent if you wanted to go into a forest and study all of the organisms in the ecosystem in a forest or in a field. It's the equivalent of sampling everything in size from an ant up to a brontosaurus, okay? So clearly there is no single net that you can take to scoop up all of these organisms. You need to develop different strategies to collect these different organisms. On Tara, we did this through size filtration, that you can see here some progressive size filtrations that we did on Tara to separate out the large organisms from the small organisms. What we also did on board Tara was to sample down to 1,000 meters in depth. Here you see how we did this using an instrument called a rosette, which has bottles loaded on it, which we can program to open and close, well, to close basically at different depths in the water column. So this allows us to take samples from as much as 1,000 meters in depth and then bring them up to the ship and analyze them. Here is a short video where you see perhaps in more detail what we do more graphically. These are plankton nets to collect larger size plankton that you see are concentrated at the end of that net, which we then scoop out. Here I think we are deploying the rosettes down into the depths of the ocean. This is sort of plankton material that we recuperate. And on board, there's a lot of filtering, a lot of concentrating of the samples in order to get the organisms onto membranes, onto filters that we can subsequently send back to the labs and analyze. We also had a small lab on board where we had some microscopes and some flow cytometers that already gave us some ideas of what sort of amazing organisms were present in the water column that we were sampling. And we also sampled at night because there are also migrations of some kinds of plankton that come up from the depth to eat the plankton on the surface during the night. Okay, so doing night and day sampling was also important. And then finally, at the end of each month or so, we would ship the samples back to the labs by cold storage to then ultimately then begin the analyses. Okay, so that's the sort of thing that we did on board. This is quite complex operations, as you can imagine. And it was also an additional level of complexity is that we wanted to do this throughout the global ocean. This is because, I mean, maybe you think of the ocean as Atlantic and Pacific and Indian and so on as separate entities, but in fact the ocean is one and everything is connected in the ocean. So even if you were a plankton, you know, sitting off Brittany today, you may find yourself off the coast of the United States or South America, or even in Antarctica sometime in the future. So we feel that the physical circulation of the oceans are likely to influence the biology of the oceans and influence the evolution of the organisms that we study. So this is why we wanted to do this sampling at really global scale, so we could compare everything with everything else. So here you see the outline of the expedition. We began in 2009, sailing from Lorient, going first into the Mediterranean, the Red Sea, the Indian Ocean, across the Cape of Good Hope, into the South Atlantic, down to the Antarctic Peninsula, around Cape Horn, into the South Pacific, out as far as Tahiti. From there, we went to Hawaii, back to San Diego, through the Panama Canal, up to New York, and then back to Lorient. And then in 2013, we did a second expedition, the Polar Circle expedition, where we went around the Arctic Circle. This is possible because Tara was built to withstand the ice, even though she's not an icebreaker. She's very happy in ice regions in cold regions. And so Tara went through the northeast passage above Russia and around and through the northwest passage above Canada in 2013, being in fact the first sailboat in history to go through the northeast and northwest passages in the same year, which is sadly not such a big thing anymore because the ice is melting in the Arctic at such a rate that it's not too difficult to do that anymore most years. So during all of this sampling campaign, we sampled at 210 sampling sites, and it was actually quite boring because we always did the same thing at every sampling site. This was very important in order to allow us to compare everything with everything else once we got the data. This was a huge undertaking because we brought back something like 40,000 samples for analysis, together with many other data sets and samples for other kinds of analyses as well. Back into the lab, we have all of these size fractions representing all of these different organisms that I introduced you to, from the smallest viruses up to the zooplankton. Using DNA and very high level, and at the genoscope in Evry. And on the other, we did a lot of imaging approaches to see what the organisms actually look like. So just to give you an idea of sort of the way we do sort of DNA-based analyses, DNA-based analyses are very powerful these days. You've seen it with coronavirus, how we can detect new variants and so on. It's incredibly powerful. It's very cheap now and very efficient. So in the case of our study, what we wanted to use genomics for was really to help us understand who is there, what do they do, with whom do they do that, and why is it important? Why is it important for them? Why is it important for the ocean, for climate? Why is it important for us eventually? Okay, so we wanted to understand this network of interactions between these invisible organisms using genomics approaches. The way typically you do that, you begin by collecting your samples, okay? It could be from a field. In our case, it was water samples from the ocean. Once you have those samples, you extract DNA. You just do a big lysis to open up all of the cells from which you extract the DNA. And then you feed that DNA into DNA sequencing machines, which typically look like these big machines here, these big boxes, okay? These sequencing machines then give you a lot of small fragments of DNA, which are just nucleotides, G's, A's, T's, and C's, organized on a string, one-dimensional, very small fragments. Once we have done quality control of those sequences, we then have to try and put those sequences together and to make some sense out of them. All of this is done computationally, really aiming first to understand what sequences came from which organisms, okay? And then once we can assign sequences to organisms, we try to understand what those sequences do, what is their function. This is what we call annotation. This is, in fact, not trivial at all, computationally very challenging to disentangle all of this, because each sample has thousands of organisms in it, many of which are unknown, and each of those organisms contains thousands of genes, okay, the vast majority of which are unknown. So it's very difficult to do that as scientists. It's sort of like doing a puzzle, doing a jigsaw puzzle, if you like, except that we don't have the drawing. We don't know what we are making when we're trying to put the image together using the different pieces of the puzzle. So that's sort of what we try to do as computational biologists. And thankfully, we've become quite good at it over several decades, thanks mainly to advances in biomedical research principally. Once we've done that, we scientists, we try to derive new knowledge. We try to understand what the sequences are telling us. We publish our data, just like you do. And then we send our DNA sequence information to public databases, which are also very well organized in the world, allowing everybody else to access our data as well. So all of this is quite well organized now, and this is what we did in Tara Oceans as well. These days, I should just tell you that these big DNA sequencing machines are now being replaced by small machines, so small that they're basically the size of a USB key. You can plug them into your computer, and you can sequence gigabases of DNA yourself at home or on a ship for a few hundred euros now. So all of this is transforming the way we do DNA sequencing, and will continue to do so in the future. So that's the DNA, the genomic side of things. What we also did is to set up approaches to actually see what the organisms look like through microscopy. I think seeing is believing when you see what the organisms look like. So this also was not trivial. We had to set up high throughput approaches to be able to do that. We did this at the EMBL in Heidelberg in Germany. And we were able through this to generate these very high resolution images of individual organisms that you see 3 examples here. For case, we can image the exoskeletons of the organisms, like in this example here, built of strontium, or this example here, built of silica. We can see the insides of the cells, in each case highlighted here in red or in blue. So we can see the internal architecture of the cells as well. And all of this information gives us insights into what these organisms do at various levels. So these are our sort of analytical tools that we were using. And obviously we have terabytes of information from DNA sequences. And we also have millions of images of individual organisms like the 3 that you see here. So it's a challenge to decomplexify all of this, but we're doing this. We've been doing this now since the expedition ended in 2013. Over the last 5 years, we have, as a consortium, we're about 20 labs worldwide, have published more than 130 papers, many of them in high-profile journals, describing often for the first time this invisible world of the plankton in the oceans. The first big publications that we made were in 2015, and I just want to go through these for a minute. These were published in Science. We were very happy that we could get a special issue of Science, much like, when there's an expedition to Mars or something, in astronomy, you will see a special issue appearing in Science or Nature. And we were happy that Science gave us this opportunity to publish our founding work from the project in a special issue in May 2015. We published 5 papers back to back in this special issue. They gave us the front cover, they gave us the editorial, and so we're very happy with that achievement. What we described in those papers were a description of the bacteria, of the viruses, and the protists in the system of the ocean. So we described, for example, a collection of 40 million genes from about 35,000 different kinds of bacteria, the majority of which were new and have unknown functions. So this is sort of the ocean microbiome, if you like. If you've heard of the human microbiome, which is associated with our intestines and our skin, this is sort of the equivalent for the ocean, containing about 40 million genes from about 35,000 different bacteria. So this is about 4 times larger than the human microbiome, in fact. So it's not so much more complex than the human microbiome. We also described about 5,500 different kinds of viruses, only 39 of which were previously known. And we described about 130,000 different types of protists, which is more than 10 times higher than the number of formally described species that we know about. And about 1/3 of them, we have absolutely no clue what they are. So those are some of our founding results. We've subsequently gone on to generate an atlas of genes from eukaryotes, from protists, from zooplankton, containing 116 million genes, which is the largest catalogue of genes from any single ecosystem on the planet, in fact, thanks to Genoscope. We've also generated genome sequences from single-celled organisms. So we can take a single cell from an organism that we don't know anything about, but which is abundant. We can pick that out, and we can now sequence this genome to tell us about what that organism actually does based on gene sequence, if it does photosynthesis, if it's a parasite, if it's a pathogen, and so on. And then we've also described, we've moved on from our initial description of 5,500 different viruses in the ocean now to 200,000 types of viruses. Okay, so this is work that we have done over the past few years. Just tell you a couple of surprises from this data. One surprise concerns, just simple things that we can address, like which is the most abundant organism in the plankton. We thought we had an idea of what that might be. We expected them to be these copepods, zooplankton, which are very easy to see when you study these plankton ecosystems. But in fact, the most abundant organisms are different. They are what we know as radiolarians, in particular 2 groups of radiolarians, which turned out to be unexpectedly abundant, and we could see those by doing sort of microscopy in situ in the water column, which reveal these delicate structures. We have known about these organisms for more than a century. These beautiful images produced by Ernst Haeckel in the end of the 19th century, in fact, stimulated the whole movement of Art Nouveau through these beautiful exoskeletons made of silica, in this case. So these were unexpectedly abundant in our data sets, and represent a major reservoir of carbon in the ocean, which we were surprised to find. The next surprise is which is the most diverse group of organisms. So it's a simple question: which group is the most diverse? And it turns out that the most diverse that we found are organisms called diplonemids. I'm sure you've never heard of them. In fact, most biologists have never heard of them either. We're again surprised to find these in our data set. This is the eukaryotic tree of life, where we have all the different kinds of eukaryotes that we know about. And we have animals here. So we human beings are sitting here in the animals, and our closest neighbor in this genealogy are the sponges, les éponges. So this gives you an idea of how complex this world of organisms is evolutionarily. And the diplonemids are sitting in this group here, which we know very little about, but we know that this group includes organisms like trypanosomes and leishmanias. So this perhaps tells us that these organisms are parasites in the ocean of other groups, but we don't know yet because we don't have them in culture. So we have a lot of work to do again to learn about what is this most diverse group of organisms in the ocean, even though we don't have them in culture yet or know very little about their biology. So with all of this data, then we have information about viruses, we have information about bacteria, we have information about protists and zooplankton. What we can then do is to take all of that data from all of those different sampling sites all over the world. we can throw all of that data together and we can see which organisms co-occur with each other. Which organisms do we always find together, or which organisms do we never find together? So this is like defining the social network of the plankton, or the ocean's Facebook, if you like. We call it the interactome. It was a very large compute done several years ago on one of the most powerful computers in Europe. And this is a result. It's not very beautiful to look at, but this is basically the network of networks of interactions of these plankton organisms. So each dot is a different organism, okay, colored by whether it's a bacteria or eukaryote or so on. And then the lines between these dots tell us if they co-occur. So green lines are co-presences, where we find them essentially always in the same sample. And then the red lines are exclusions, where we never find associations between the organisms, okay, in our sample. So here we begin to see this huge amounts of information in here, but on a broad level, we can see that there are some organisms that are very sort of social. They have lots of interactions with organisms around them, lots of green lines. These are sort of I don't know, the Jennifer Lopez, the Beyoncés of the plankton that are very popular, the Pogbas, if you like. And then we have these plankton which are very negative, which exclude other groups of plankton. So some of them over here are very, very antisocial, very unpopular, if you like. These, perhaps we can call them the Trumps, the Putins of the plankton world. We also have these isolated sects of plankton which sort of keep to themselves. And so all of this is a lot of information to help us understand how the organisms interact with each other. And one thing that we were surprised to find from this information is that the biology seems to be particularly important. So when we compare this with the interactions between the organisms and the interactions between the organisms and the environment, like temperature or salinity or something, we see that the biology is more important than the environment. So basically, it's more important who you live with than where you live if you're a plankton. Which again, we were quite surprised to find in our data. So what this data also is, it's a sort of playground to discover interactions between organisms. So you can take your favorite organism, if you have one, and you can see who it interacts with. This is, of course, computational output from DNA analysis, but we can also go into our imaging data set and see if we can confirm those interactions. And here are a couple of examples of that, where we have a diatom chain here, which has silica exoskeleton interacting with an organism called Vorticella. And you can see that this is a parasite, probably. You can see this single cell parasite, particularly nasty, sticking to this chain of diatoms and presumably infecting it. What we also can see from these interactions are symbiotic interactions. This is an example of a diatom chain with 6 diatom cells in it. And inside each of these diatom cells, we see these green balls, which we have labeled, which are nitrogen-fixing bacteria. So these are just like the kinds of bacteria that associate with some plants, leguminous plants, and fix nitrogen on land. And we know of the importance of nitrogen fixation in agriculture, in forests, for bringing nitrogen gas into a form that can be used by biology. We know that it is at least as important in the ocean as it is on land, but it's difficult to study because these organisms are microscopic. And here, so we have a way into this world of nitrogen fixation in the ocean to learn more about who does it and where do they do it and its importance and so on. So those are the sorts of things that we can do by looking into this data set. Now, I want to zoom out a little bit and look at photosynthesis on our planet. And this is satellite image, or composite satellite image, which we can take from satellites which are able to detect chlorophyll on the surface of our planet. I'll talk more about this a little bit later. And so by measuring chlorophyll on the surface of our planet, we can see that the chlorophyll in the oceans is in fact as important as the chlorophyll on the land. So the photosynthesis in the ocean is at least as important as the photosynthesis in the tropical forests, in the fields, and so on land. So this was quite a surprise when we first started viewing our planet from space and looking specifically at the chlorophyll. You also see how dynamic it is. It reflects the seasons. You can see on land the desert greening up during springtime. And also in the ocean, you can see a lot of dynamics. This, for example, event you're going to see here with a big algal bloom over the Pacific is related to El Nino, La Nina transitions in the Pacific of ocean warming. So this is our view from space of photosynthesis. Our view down the microscope of photosynthesis looks very different. Looks something like this. So as I said, we have very few plants that we can see in the ocean. We have some seaweeds, but they're not so important globally for photosynthesis. The plants that do photosynthesis in the ocean are essentially all microscopic. And here are some images of what they look like. We have bacteria, very small bacteria that do photosynthesis. And we have other kinds of eukaryotic phytoplankton that does photosynthesis, including green algae, you can see here, or dinoflagellates here, which have organic exoskeletons, very elaborate. We also have diatoms, which have silica cell walls. You can see here, and I've showed you before. And then we also have organisms that can manipulate calcaire, calcium carbonate, and produce these beautiful exoskeletons made of calcium carbonate. And these can deposit over many millions of years and make the sedimentary rocks that we see all over the world. And then we also have symbiotic organisms where we have algal symbionts living inside other organisms as well. So this is our view sort of from the microscope. What we can do using the Tara Oceans data set is try to get a composite view of which organisms do what in the global ocean. And so here we see all of these different groups, how they're distributed in the global ocean. And to do this, we're using a universal marker, a marker gene, which is present in all photosynthetic organisms, be it tiny bacteria or the larger eukaryotes like diatoms and dinoflagellates. So here in this one single image, we can see sort of the abundance of each of these organisms, the relative abundance of each of these kinds of organisms. These are diatoms. And we see their abundance by the size of the circles is particularly strong in Arctic and Antarctic, and less so around tropical regions. Whereas the photosynthetic bacteria, like this Prochlorococcus, is much more in tropical lower latitude regions. Okay, so this gives us a view of all of these different kinds of organisms doing photosynthesis. And we can see their relative importance in different regions of the ocean. We can also look at their diversity, and this I've shown using this index called Shannon index, and basically red colors are highly diverse populations of particular organisms, whereas blue-green are least diverse groups of populations of organisms. So again, we can see how diversity changes around the planet, and in particular that diversity is richest in tropical regions, and then diversity gets reduced as you go towards high latitude. So it's exactly as what we see on land in terms of latitudinal gradients of diversity, we can also see in the ocean. So this is sort of the first time we're able to view all of these different organisms at the same time using one single approach. What we can then do beyond just sort of looking at their biogeography is to look at their function in terms of photosynthesis. So which group of organisms is the most important contributor to photosynthesis in the ocean? So here we have a heat map clustering analysis where we have different organismal groups. So basically the biology, different biological groups of organisms along this axis. And we're comparing this with different environmental parameters, which we collected as well during the expedition. And in terms of photosynthesis, what is important, what are the important parameters are net primary production. This we can calculate from space, from the satellite data. And then we can also compute chlorophyll concentrations by measurements actually in the water column to see how much chlorophyll is present. And so we see here the association of these photosynthetic parameters with all of these different groups of biological organisms. And the group that is the most strongly then correlating with photosynthetic activity in terms of primary production or in terms of chlorophyll in the water are the diatoms here. Okay, so that was quite, we sort of suspected that diatoms are important in the ocean, but here we see very clearly that the diatoms are particularly important for contributing to photosynthesis in the ocean. So photosynthesis is the way that carbon comes into the ocean biologically. Carbon dioxide from the atmosphere is converted into organic material by these photosynthetic organisms, which then enters the food chain. And this is part of what we call the biological carbon pump in the ocean. But it's only one half of the carbon pump, because the other half is the sinking of this material to the bottom of the ocean, because the ocean is deep, because we have gravity on our planet, particles sink to the bottom of the ocean. So this is a very slow process of all of the carbon that is taken up by biology in the ocean. Only perhaps 0.1% of it will actually make it to the bottom of the ocean. But this over geological time creates huge reserves of carbon in the form of oil and gas. So oil and gas that we're burning today is just simply the remains of plankton, which have sunk to the bottom of the oceans over huge geological times of tens of millions of years. So this is very important for climate regulation, as you can appreciate. But this is quite difficult to study because the ocean is deep and it's difficult to see actually these particles sinking to the bottom of the ocean. What we can do in the Tyra Oceans data set to address this is, on the one hand, look at the data we have from particulate material in the ocean and derive carbon flux estimates from the global ocean that you see in this map. Here we have sort of particulate material in the ocean down to depths of 500 meters. You can see that it's not at all homogeneous. You have some areas that are sort of oceanic deserts, like the South Atlantic. You have other areas very active in carbon export, like the Southern Ocean, you see here. So we have this data, and we have, all this interactome data that I showed you before. So what we can do using machine learning approaches is combine these data sets and sort of go from a rudimentary view of plankton communities to a more graph-based view to sort of identify which organisms are the most important in the network as being associated with the carbon export that we observe at different sites. So here we can identify specific organisms which appear to be particularly important in this process. We can imagine if those organisms are sensitive to climate change and would disappear, that the whole network associated with carbon export could actually break down So identifying this information and the organisms that are hubs within this network is particularly important. And what we identified from this study are organisms that we expected to identify, like the radiolarians and dinoflagellates who've been associated with carbon export in the past. But we were also very surprised to find little guys like the bacteria, photosynthetic bacteria, in the group of Synechococcus, and even the viruses that infect Synechococcus, that are known as synechophages. These appear to be very strongly associated and central to this network that we identified as being associated with carbon export. So even the small organisms, small life forms appear to contribute to this process, which was a surprise for us to find. So to summarize this major part of my talk then, I've prepared this summary slide where you can see what different things that I tried to talk about. Tara Oceans, I think, is most significant because it is the first end-to-end description of a continuous global ecosystem. End-to-end because it's from viruses all the way up to zooplankton, so covering the whole food chain, and continuous global ecosystem because it's the ocean sampled at a global scale. So it's really the first example of that we have from scientific studies. The ocean microbiome, the bacteria in particular, consists of about 35,000 different kind of species of bacteria, containing around 40 million genes that are mostly unknown. The eukaryotic plankton, the protists, the zooplankton, the diversity is huge, about 130,000 different types, but it is finite. This is sort of, the more now we sample, the more we just find the same thing. So this is sort of a reasonable estimate for the number of organisms, different organisms that we have in the ocean concerning the protists and the zooplankton. However, about 90% of these are new, and for about 1/3 of them, we have absolutely no clue what they are. So, for example, the diplonemids, we still struggle to understand what they're doing in the ocean. We've also described a catalogue of genes from these organisms that is now 116 million, which is a great resource for people to look at and learn more about what these organisms do. And in terms of the viruses, we now have described about 200,000 viral types, of which more than 99% are new. So again, we need to understand what these viruses are doing in the environment, who do they infect, how do they infect, and so on. I showed you quickly the plankton interactome, revealing the importance of the interactions between the organisms in particular. And I gave you some feel for how we can use the microscopy combined with the genomics to tell us more about what these organisms do. And the example I gave you was concerning the biological carbon pump, the input of carbon into the ocean through biology via photosynthesis, and the export through carbon export. And all of this data is public, so other people are as welcome to use the data as we are, and in fact are doing so. So ultimately, this data set allows study at different levels, people who are maybe just interested in a DNA level, interested in metabolism, how organisms work, through to population level, through ecosystem and whole planetary level. So it provides different opportunities to explore the ocean in different ways. And in particular, looking at the interface between these biological systems and the functioning of the Earth system as a whole, and the feedbacks between them. And I just wanted to share some more preliminary data with you in the last few minutes of my talk, if I can, which is maybe the part that will interest you the most. So if you've been sleeping up till now, please wake up, because here I'm going to talk about satellites, satellite imaging, in particular chlorophyll, and our attempts to link this to genes in the ocean that we're looking at through genomics technologies. So this is quite a challenge, as you can imagine. I'm very proud that we have a postdoc funded by the CNES in the EBENS to study this, whose name is Roy El-Hurani. And he's basically developing new machine learning approaches to try to observe the diversity of photosynthetic organisms from satellites in space. Okay, so I just want to spend a few minutes talking about this before we break for questions. So imaging chlorophyll from space, we've been doing it since 1978. The first satellite was a NASA satellite called CWIFS. Here you see the first available image that Roy could find from 1st of November 1978, where we can see chlorophyll detected spectrally from the satellite. The way that this works is essentially measuring, spectra, as in all, satellite imaging, in particular looking at blue, red, and green wavelengths. And because chlorophyll has differential absorption at these wavelengths, we're able to backtrack and calculate chlorophyll from the spectral data. But we've gone a lot beyond that these days, and we're able to do more with satellite data, which I can show you in this slide here. You can see, we can look at different spectral properties of the surface ocean and define different things. So on the left, you have a standard chlorophyll image of the United Kingdom and Northern Europe. But you can see different features in this image. You see some areas with, that have different properties. Okay, so in each of these areas, you can see that on the left, we may have, just a scale bar of chlorophyll concentrations, whereas on the right, we have different spectral features that we can try to use to try to see which organisms are actually present in these different regions. So we've been trying to do this for some time. Here is a summary of different publications that have already emerged from this sort of study, basically using spectral anomalies from satellite data to try to learn more about the organisms in the ocean. So looking at these different phytoplankton groups, we can try to define them biogeochemically. So which ones make calcium carbonate structures, which ones make silica structures, which ones don't make any structures, for example, which ones sink through carbon export, which ones sink less. We can also be interested in the size class of the organisms, because some are small and some are large, which can make differences for carbon export. And then we can look at the distribution of particle size in the water. So all of these parameters have been attempted to be defined using, based on the spectral anomalies observed from space, be it radiance-based, absorption-based, scattering-based, and so on. And this has made some progress in the field. And what now is interesting to do is to see if we can make these relations even stronger by sort of going beyond chlorophyll and use the spectral anomalies of the different pigments that are present in these different kinds of photosynthetic organisms. So here we have spectra of the different pigments that are present in different kinds of plankton, different kinds of phytoplankton. So they all have different spectra. The diatoms are characterized by fucoxanthin as an additional pigment in addition to chlorophyll. Some of the cyanobacteria, the prochlorophytes, have other forms that we can detect. And we typically would detect these through pigment analysis of water samples through HPLC kinds of approaches. So what Roy has done during his PhD and now in my postdoc is sort of using neural network classifications of chlorophyll data from space and spectral anomalies detected from space, and connecting that up with the concentrations of these secondary photosynthetic pigments that are specific for different kinds of photosynthetic organisms. For example, fucoxanthin for the diatom. So we have large data sets publicly available of these secondary pigments from multiple expeditions done in the past. So Roy took that data and was able then, using the satellite data combined with the secondary pigment analysis, to get, for example, distribution data of, in this case, diatoms, looking at fucoxanthin concentrations and comparing this with the spectral anomalies from space. So here we get more precise distribution data for particular groups of photosynthetic plankton. So this then allows you to go back to the satellite data, the chlorophyll data, a composite image here over time, and to break this down into which different organisms are doing photosynthesis in each region. Okay, so here we have this is shown by the color bar of the cyanobacteria are shown in yellow here. The diatoms are shown in orange, for example. And so this allows us to sort of disentangle the chlorophyll data into the photosynthetic groups of organisms that are doing the photosynthesis in a particular location. The problem is that life is more complex than that, that the photosynthesis in a particular region is not just defined by one group of organisms, be it diatoms in orange or cyanobacteria in yellow, but they're all present at the same time, right? So we need to be able to try to incorporate all of that data together to help us get a better view of who is doing photosynthesis in a particular area. So Roy has taken this a step further, and as beginning with the satellite data, the neural network classification, combining with these secondary pigments that are diagnostic of these different photosynthetic plankton groups, you can now combine this with the genetic data that we're getting, for example, from this PSBO gene, where we can quantify each of these different phytoplankton groups at the same time. So this PSBO data set from the genomics data of Tara oceans can then be used to sort of train the satellites to tell us who is where. And here are composite maps where we have diatom, green algal, dinoflagellate, and so on distributions, as viewed with the help of PSBO genetic data, but essentially observed from space using spectral anomalies. And you can see that we're able to break it down quite well into these different groups. And we can see, you know, not only the single dominant type of plankton, but also how different types of plankton are grouping together, are assembling in different regions. So this is sort of the first time that anybody has been able to sort of go from the gene level up to the satellite level. It's still early days with this analysis, but I wanted to share it with you because perhaps some of you are interested to be involved in this work, or you have advice to help us as well. So I will end with that more forward-looking story of connecting genes with satellites, which can have great interest for space, for satellite imaging of the planet, because it can save us a lot of time and a lot of money, potentially, if we're able to disentangle the photosynthetic groups that are doing photosynthesis in the ocean from satellite data. It gives us the spatial resolution, it gives us the temporal resolution of daily images and so on, which would be impossible to achieve if we are going out on boats and doing metagenomics every day and so on. So this is a promising new approach, which NASA, which ESA are very interested to follow up with us in using the genetic information to disentangle the anomalies viewed from space. So I will end with that story, and let me thank the people involved in the work, which are numerous people. We have different institutes involved in this project, French, but also international. We have different funders who have funded the research side of the project, and also different sponsors who funded the expedition side, in particular the Tara Ocean Foundation. So I will end there and thank them, and thank you for your attention as well. Okay.
Thank you very much, Chris, for this great presentation. So we have time for questions. Please, si vous avez des questions.
I have one. So I'm Daniel Rouen from the Observatoire. So thank you. Very, very interesting. Probably I will have to replay the record because I didn't catch all the details, but anyway. So now my question. So you mentioned the very important role of bacteria, but you never mentioned, as far as I have seen, the second class of prokaryote and the archaea. And so what does this mean? That they are not present in the ocean, or they do not play a role in photosynthesis? So please.
No, the archaea are certainly present in the ocean, but archaea tend to live in more extreme environments, be it high temperatures or high salinity, low oxygen environments as well, anaerobic environments. And because we were largely sampling the aerobic surface ocean, or the top 1,000 meters of the ocean, there are not so many archaea present in these environments, but they are present in our data sets, certainly. Yeah.
Alors, je vois qu'il y a une question de Thibault Lebertre. Alors, je peux la lire. Comment se compare la quantité de carbone dans le plancton et celle correspondant au CO2 dissous dans l'eau de mer? Question dans le chat.
The biological carbon pump is only one process whereby CO2 can enter into the ocean. The stronger component is the physical pump, the physico-chemical pump, which just through physico-chemical processes dissolves, mixes carbon dioxide with the water, generates carbonate, bicarbonate in the ocean. Is one reason why the oceans are acidifying. And so this is more, I can't remember completely well the relative importance of each, but I think it's maybe something like the physico-chemical pump is about 90% of the carbon that enters into the ocean, whereas the biological pump is maybe 10% or something like that. So it's not so important that, you know, the photosynthesis is clearly less important than the physico-chemical process of carbon uptake into the ocean. But the difference, I think, that is important is that the biological process allows the removal of carbon dioxide out of the carbon cycle through the deposition of carbon to the bottom of the ocean in the form of organic material, which can be removed from the active carbon cycle for geological periods of time. And equally so, the organisms that use carbon dioxide to make calcium carbonate, calcaire, these organisms also then can sequester the carbon dioxide from the carbon cycle and put it into calcium carbonate, which is essentially inert to biological recycling. So these are the ways how the biology in the ocean impact the carbon cycle in very profound geochemical ways, in fact. I hope that answers the question.
Thank you. D'autres questions, other questions? In fact, I have one. It may be more related to the next slides in your presentation that I had a look at before. And I think you mentioned that during your talk, that there is a gradient of biodiversity between the poles and the equator, with a maximum at the tropics, like on land. And I was, in fact, very surprised about that, because I read that the tropical seas are relatively dead, not relatively poor in nutrients, that correspond to downwelling region in the ocean. And when we look at a map of net primary productivity of or chlorophyll, as you show in your movie, there is a maximum essentially at the equator and at high latitudes. So is it a distinction between biodiversity and biomass, or am I missing something else?
Yes, it's so we're really only looking at biodiversity and not biomass. That's right. And in this slide here, we look at those diversity gradients by latitude, from Arctic through tropical regions through Antarctic, and we have the different groups of organisms, be they prokaryotic bacteria, archaea. So here we have archaea, for example, that is also in this plot, and you see that in basically every case we have a latitudinal gradient of diversity. So diversity is highest in the tropical regions, which is also the higher temperature regions. Although the relation is quite more complicated if we look at productivity or biomass in terms of chlorophyll concentration. Chlorophyll is higher at the higher latitudes, of course. Okay, so there are complex relations between the patterns of biodiversity that we see and the temperature and the productivity of the system. So, in fact, low nutrient environments have low biomass, but they often have they're often very rich in diversity because of many organisms that are present. They are present in very low numbers, but they are present in large diversity. So this is quite interesting to see that. It's similar to what we have on land, of course, in terms of gradients being strongest at the tropics, and being certainly driven by temperature, but it's not only temperature that is important for the patterns that we observe. Voila.
Thank you. Other questions?
I have a question. Based on not only the data from Tara, but from other sources of data, I guess, over the last decade or a few decades, do we see any time evolution for the planktons, or one of the categories that you have mentioned, like the diatoms, for instance, which seems to be important for the photosynthesis?
Yes, so we cannot really address this with Tara data in any very, I think, serious way, because Tara is really giving us a snapshot of the ocean at the beginning of the 21st century. We would have to repeat Tara again in the future decades to see trends, changes happening eventually. So we cannot really say much about that with our data set. There are other data sets that show, of course, the temperature of the ocean is increasing, and there's data showing organisms are migrating. So when they can, like the copepods, the zooplankton that can move, they are migrating. The fish, certain fish stocks are following the plankton because they eat them. So there are changes that are happening coming from other data sets. The kinds of things that we can do is to take our data, for example, of diversity gradients of plankton, and inject them into the model ocean of from the GX, from the IPCC, where we have projected sea surface temperature change, change in primary productivity in the ocean by the year 2100. We can put our plankton distributions into these virtual oceans and see how they repartition, and this sort of gives us a view of kind of diversity anomalies that we predict in the ocean by end of 21st century. And so for copepods, for example, the zooplankton, we predict decrease in diversity in tropical regions and increases as they can explore the Arctic environment in more abundance because of the melting of the Arctic region. And by looking at these diversity maps, we can see how that maybe is going to influence important features of the ocean. For example, the carbon export, the industry of fishing, and the efforts of marine conservation through design of marine protected areas, which we can see on these map monde. The sort of general view of carbon export in the ocean through biology, the annual fish industry are shown here, and the density of the marine protected areas, and the diversity that we predict in the future ocean of the plankton at these higher latitudes is likely to impact all of these processes, in fact. So these are sorts of ways that we can try to use the Tara data to project how these organisms may be sensitive to changing temperature changes, etc. in the ocean. So these are ways that we can try to do that. And we can also use other methods to try to see maybe how these plankton have changed over previous time in the last tens of billions of years, for example, using other approaches. So there are ways that we can do this, but the best way would be to go and repeat the project in 10 years' time or something.
Thank you.
That was a bit long.
Thank you.
Other questions? In fact, I have one related to climate projections. Plankton is an important source of aerosols for cloud condensation nuclei by the dimethyl sulfide. Is it something studied, how it would evolve in the future and how it could impact the climate?
Yes, certainly. If we can get a more accurate idea of which kinds of plankton are producing the dimethyl sulfoxide, the cloud condensation nuclei, yes, for sure, we can then try to see their distribution today and project their distribution into the future using, for example, the climate models from the IPCC. So yes, that is something that we can do. We have now good information of which genes are responsible for making the DMS in the cells. So we can do a analysis of which kinds of plankton can make DMS from analysis of their genetic potential. And then we can see how those organisms are distributed today and how they may change their distribution in the future. So yeah, it would be something interesting to do. I don't think anybody is doing it, but it would be interesting to try.
No? So in that case, I suggest that we applaud Chris for this great presentation.
Thank you.
Il a allumé vos caméras pour applaudir.
Benjamin, j'ai peut-être juste une question technique. D'abord, merci beaucoup, c'était vraiment fabuleux. Je n'ai pas de questions intelligentes à poser, mais je regarderai certainement encore la vidéo pour apprendre d'autres choses. Est-ce que vous autorisez que l'on mette le lien vers l'enregistrement sur un site qui est en libre accès, qui n'est pas limité au groupe, mais
oui, en principe, il n'y a pas de problème. Je dois juste vérifier avec mon postdoc, Roy El-Urani, qu'il est bien aussi avec ça. Mais en principe, c'est OK, je pense.
D'accord, donc j'attends que